In order to make proteins, DNA must be translated into RNA. Mutations can and often do occur at the DNA level that can change proteins, making them non-functional or sometimes having no effect. A point mutation results in the substitution of one base for another. 4 types of point mutations include a missense mutation, nonsense mutation, silent mutation, and a read-through mutation. Let's dive into what they are and how they affect us. Here we have a coding strand of DNA. We can translate this into mRNA that code for amino acids in order to make proteins. If a single base nucleotide is mutated, it can change the amino acid and change the protein. In this example, the nucleotide base A is mutated to G, which causes the amino acid to change from asparagine to aspartic acid. This is an example of a missense mutation, which is when the amino acid is changed and may alter the protein function. This is also an example of a transition because the nucleotide base changes from A to G, which is purine to purine. The next example is a mutation of C to A nucleotide bases. In this case, the amino acid changes from cytosine to a stop codon. This causes the protein to stop earlier than expected and alters the protein function. This is considered a nonsense mutation, which is when a codon changes so that it becomes a stop codon. The change from C to A makes it a transversion, pyrimidine to purine. The next example shows a mutation from U to G. This actually does not have an effect on the protein because they code for the same amino acid. This is known as a silent mutation and creates the same protein for original function. The U to G mutation is a transversion because the original DNA was T instead of U, therefore is considered a change from a pyrimidine to a purine. The last example I have shows a mutation from U to A, causing the original stop codon to become an arginine. This change is called a read-through mutation, where a stop codon is changed to a codon that codes for an amino acid. This creates a longer protein and therefore alters protein function. The change from U to A is similar to the previous example as to why it is a transversion. Sickle cell anemia is a disease caused by a point mutation of A to T. The amino acid changes from glutamine to valine, making it a real-life example of a missense mutation.